Hello everyone. Today we are going to discuss about beta oxidation of fatty acids. So when does this beta oxidation occur? So beta oxidation occur mainly during the starvation state. So what happens during starvation? We know that our body is devoid of energy. So what happens? The insulin, that is decreased insulin, increased glucagon ratio. So decreased insulin and increased glucagon ratio, it favors what? Lipolysis. That is the lipolysis in the adipocytes. So in the adipocytes, the storage form of fat is triacylglycerol. So that is being broken down to form the fatty acids. And this fatty acids undergo oxidation in the mitochondria of liver. Mitochondria of liver, and we get the acetyl-CoA. So that is how beta oxidation occurs. So first we will see its definition. Definition of beta oxidation. Then the steps involved in the beta oxidation. Then its energetics. How much energy we are getting. Then its clinical applications. And also the regulation. So first we will see the definition. So as the name implies, beta oxidation. So beta oxidation, it means the oxidation occurs in the beta carbon atom. So if you are taking a long chain fatty acids, the oxidation occurs in between the alpha and the beta carbon atom. Okay, the carbon atom carrying the carbonyl is the alpha carbon. So beta oxidation occurs in between the alpha and the beta carbon atom. So the long chain fatty acids is broken down in the form of the 2-carbon compound acetyl-CoA. So that is how it's breaking down the fatty acids. Okay. So that is the definition. That is, oxidation occurs in between the alpha and the beta carbon atom. Now we will see the steps involved in the beta oxidation. There are 3 major steps involved. First is the activation. The fatty acids is activated to a high energy compound. Second is the transport. So this is important in the case of long chain fatty acids. The long chain fatty acids, because the activation occurs in the cytoplasm, it has to enter the mitochondria. So long chain fatty acids require a transporter with the help of carnitine. Carnitine is a transporter. And third is the proper beta oxidation steps which occur in the mitochondria of liver. Okay, the steps of beta oxidation. So these are the 3 major steps involved. So first we will see the activation. So beta oxidation first is activated to its acyl-CoA with the help of thiokinase. So the fatty acid with help of thiokinase, it forms fatty acyl-CoA, which occurs in the cytoplasm. And for this activation, 2 high energy bonds of ATP is used. So ATP is converted to AMP and phosphate. Okay. So that is the first step. The activation of fatty acids occurs in the cytoplasm. This occurs in the cytoplasm. The second is the transport. So the short chain and the medium chain fatty acids, they can traverse the mitochondrial membrane easily. But the long chain fatty acids require a transporter, and that transporter name is carnitine. So carnitine for the transport of fatty acids from the cytoplasm to the mitochondria. So mitochondria is the place where the beta oxidation, the steps of beta oxidation occurs. So carnitine is the transporter. So what happens is, so from the cytoplasm, it has to traverse the mitochondrial membrane. So it has the outer mitochondrial membrane and the inner mitochondrial membrane. So a fatty acid has to reach from the cytoplasm to the inner mitochondrial membrane. So the outside of the outer mitochondrial membrane, we have an enzyme, that is a carnitine acyl transferase, CAT. Okay, carnitine acyl transferase, that is for the transfer of acyl group. If we are transporting a palmitic acid, then we will say as carnitine palmitoyl transferase. So this is the CAT1 that is present in the outer mitochondrial membrane. What does it do is the carnitine will accept the acyl group to it. That is our fatty acyl CoA will become acyl carnitine. That is acyl group. The fatty acyl group is transported to the carnitine. So this acyl carnitine traverses the translocase through the mitochondrial membrane. So for that we require an enzyme translocase. So translocase helps in the transport of acyl carnitine from the outer mitochondrial membrane to the inner mitochondrial membrane. So in the inner mitochondrial membrane we have another enzyme, that is CAT2. That is a carnitine acyl transferase 2 or the carnitine palmitoyl transferase 2 if you are transferring the palmitic acid. So what does it do? The acyl carnitine, it releases the carnitine and the fatty acyl CoA is regenerated. Okay, so that is acyl carnitine liberates carnitine. Carnitine goes back and what we get is a fatty acyl CoA. Thus our long chain fatty acyl CoA reaches inside the mitochondria. So mitochondria is a proper beta oxidation steps occur in the mitochondria. Now our fatty acyl CoA has reached inside the mitochondria. So now I will see the steps involved. So inside the mitochondria, so if you consider mitochondria as one hot boiling system, we can remember the steps of beta oxidation. That is O hot. O stands for oxidation, H for hydration, again oxidation and then thiolase, the breaking down. So first step is oxidation. So the fatty acyl CoA, that is if you consider that as a palmitic acid, this fatty acyl CoA will undergo oxidation. Oxidation means it's the removal of hydrogen atom. It's removed from the alpha and the beta carbon atom. So two hydrogen atoms are removed and FAD is the acceptor of hydrogen. The first step, F stands for F and FAD will become FADH2. That is hydrogen atoms are transferred to the FAD and it becomes FADH2 and we get a compound. That is a enoyl CoA. That is a trans-enoyl CoA. So you will get a trans-enoyl CoA and this FADH2 goes to the ETC, electron transport chain, and we generate 1.5 ATP. Okay. Then this trans-enoyl CoA undergoes hydration. That is a water molecule is added in between the alpha and the beta. And we get beta hydroxy fatty acyl CoA. So that is a hydroxyl group in the beta position. So beta hydroxy fatty acyl CoA. Then it again undergoes oxidation. There is again two hydrogen atoms is removed from the beta carbon and what we get is beta keto fatty acyl CoA. Beta keto fatty acyl CoA. And finally P stands for thiolase. That is it's broken down in between the alpha and the beta. And what we get is the CH3COSCOA. That is acetyl CoA and this will again undergo beta oxidation. So the long chain fatty acids. So what we will get is that is another fatty acyl CoA and plus acetyl CoA. That is, if you are considering a long carbon atom like a palmitic acid, 16 carbon palmitic acid, what happens is after one cycle of beta oxidation, we will get a 14 carbon fatty acid, fatty acyl CoA and acetyl CoA. Thus, for the complete breakdown of 16 carbon atom fatty palmitic acid, we require how many cycles of beta oxidation? Yes, we require 7 cycles of beta oxidation, which generates 8 acetyl CoA. Okay, so if you consider that is N by 2 minus 1 cycles of beta oxidation and N by 2 acetyl CoA. Okay, so these are the steps involved in the beta oxidation. That is, first the fatty acyl CoA undergoes oxidation and it forms a transsinoyl CoA. Then it again undergoes hydration and it forms beta hydroxy fatty acyl CoA. Then it again undergoes oxidation, and in the second oxidation NAD is the acceptor of hydrogen. So NAD will become NADH plus H plus. And this NADH will enter the ETC to form 2.5 ATP. So in the first step FADH2 is generated, which goes to the electron transport chain and we will get 1.5 ATP. The third step NADH is generated and it goes to the electron transport chain to generate 2.5 ATPs. Then the breakdown occurs and it forms a fatty acyl CoA and acetyl CoA. Okay, this is a one cycle of beta oxidation, and for the complete oxidation of the 16 carbon atom palmitic acid, we require 7 cycles of beta oxidation. So we require 7 cycles of beta oxidation. That is, for 16 carbon atom palmitic acid, we require 7 cycles of beta oxidation. So 7 cycles of beta oxidation will generate 8 acetyl CoA, and each cycle generates 1 FADH2. So 7 FADH2, 7 NADH. Okay, so what will be the energetics? So 8 acetyl CoA, and these acetyl CoA, one acetyl CoA enters the TCA cycle and generates 10 ATP. One acetyl CoA enters the TCA cycle and generates 10 ATP. That is acetyl CoA combining with oxalate state forming citrate. The cycle continues and each cycle of TCA generates 10 ATP. So 8 acetyl CoA means it generates 80 ATP, and 7 FADH2. It generates 7 into 1.5. Right? So that is 10.5 ATPs and 7 FADH2. So that means 7 into 2.5. It generates 17.5. So total 80 plus 10.5 plus 17.5, 108 ATPs is generated. And for the initial activation of beta oxidation, we are using 2 inorganic phosphates. So the net ATP generation in the beta oxidation is 108 minus 2. The 2 inorganic phosphates we use for initial activation, and it forms 106 ATPs. Okay, so that is energy generation in the case of palmitic acid. So if you are considering it as an 18 carbon stearic acid, then how many cycles of beta oxidation will occur? That is 18 by 2 minus 1. That is 8 cycles of beta oxidation. Then we will get N by 2. That is 18 by 2 is 9. 9 acetyl CoA. And the same energy calculation we have to do. Okay. So that is it for any number of, especially for long chain fatty acids, we will use this formula. That is N by 2 minus 1 cycles of beta oxidation and N by 2 acetyl CoA generation. Fine. So that is about the energetics. Now we will see the regulation. How is the beta oxidation regulated? So the major entry point in the beta oxidation is our transporter, especially in the long chain fatty acid. They require the transporter. Carnitine acyl transferase 1. Okay, that is CPT1. So this is the major regulator. Okay, so major regulator is the CAT1. So the glucagon, the hormone, all favors the activates CAT1. CAT1. Okay. And the malonyl CoA, in order to prevent beta oxidation and fatty acids to occur simultaneously, what happens, the malonyl CoA, which is the initial compound formed by the fatty acid synthesis, it is an inhibitor of CAT1. Inhibits cathode. Okay. So that is about the regulation. Then the clinical applications. So clinical applications of the beta oxidation, mainly the most important clinical application is the medium chain acyl-CoA dehydrogenase deficiency, MCAD deficiency. So this is especially important in case of babies. Why? Because breast milk is rich in medium chain fatty acids, lauric and myristic acids. So if this enzyme is deficient, the medium chain acyl-CoA dehydrogenase deficient, what will happen? Beta oxidation will not occur. So decreased beta oxidation, especially if we are not feeding babies at night, that is prolonged fasting. Prolonged fasting will result in decreased beta oxidation. So if beta oxidation is decreased, what will happen? There is decreased acetyl-CoA. And we know that acetyl-CoA is an activator of pyruvate carboxylase. So pyruvate carboxylase, what is the role of pyruvate carboxylase? It converts pyruvate to acetyl oxaloacetate. Okay, so if this cycle will be blocked, that means gluconeogenesis is blocked. Also we know that one cycle of beta oxidation, that is in case of palmitic acid, generates around 106 ATPs. So decreased ATP, so gluconeogenesis will not occur. So these two results will result in fasting hypoglycemia. Okay, especially if the babies are not fed at night, they are not fed at night, what will happen? There will be fasting hypoglycemia because of decreased gluconeogenesis. Okay, so MCAD deficiency is one important clinical manifestation, and that can result in sudden infant death syndrome, SIDS, sudden infant death syndrome, also called crib death. So babies with MCAD, medium chain acyl-CoA dehydrogenase deficiency, if they are not fed at night or if there is prolonged fasting, can result in the death of the babies due to the decreased gluconeogenesis. Then another important clinical application is Jamaican vomiting sickness. Jamaican vomiting sickness. So in Jamaica, it's a place in Africa, there is a fruit, particular type of fruit called as ackee fruit. So those who ingested this fruit, ackee fruit, it contains a toxin, that is hypoglycin, which inhibits the acyl-CoA dehydrogenase enzyme. Acyl-CoA dehydrogenase enzyme. So beta oxidation will not occur. So decreased beta oxidation. So decreased beta oxidation means gluconeogenesis will not occur. So decreased gluconeogenesis. So that will result in fasting hypoglycemia. And the patient will present with continuous vomiting and even convulsions can occur. But there is no ketone body generation. Why? Because decreased beta oxidation. So decreased acyl-CoA. Acyl-CoA is not available for ketone body formation. So no ketone body formation. So in Jamaican vomiting sickness, there will be there will be hypoglycemia and there will be no ketone body and the patient presents with continuous vomiting. So these two are some significant clinical applications of beta oxidation. Also, if the enzymes are deficient, the carnitine acyltransferase or the carnitine acyltransferase 1 or 2, or if carnitine is deficient, that all will affect beta oxidation. This carnitine especially is important in muscles. So deficiency of carnitine can result in the muscle cramps and also decreased beta oxidation. So these are some of the important points in the beta oxidation. So to sum up, we can know that beta oxidation, the oxidation occurs in between the alpha and the beta carbon atom, and the beta oxidation mainly occurs in the liver mitochondria. Okay. And the three major steps involved is activation of fatty acid which occurs in the cytoplasm. Then the transport is required for long chain fatty acids. Short and medium chain fatty acids do not require a transporter. And carnitine is a transporter of beta oxidation. We have carnitine acyltransferase 1 and carnitine acyltransferase 2. So with the help of carnitine, the long chain fatty acids enter inside the mitochondria, and inside the mitochondria the four major steps of beta oxidation occur. That is oxidation, hydration, oxidation and catalysis. So that will break down, and each cycle of beta oxidation releases 2 carbon acetyl-CoA. Okay, suppose 16 carbon atom palmitic acid will require 7 cycles of beta oxidation and it generates 8 acetyl-CoA. So regarding the energy formation, each acetyl CoA will form 10 ATP, each FADH2 will generate 1.5 and each NADH2 will generate 2.5 ATPs. Okay. So that is about energetics, and regarding the clinical application, most important clinical application is the MCAD deficiency, that is a medium chain acyl CoA dehydrogenase deficiency, and also Jamaican vomiting sickness. I hope this topic is clear for you. Thank you.